Great. Thank you, Scott, for that introduction. Appreciate being invited here to speak with all of you today. And thank you for taking the time to be here. Today, I'm here to talk about one of our arms of research at Persephone Biosciences. There's a few things that we're interested in, microbiome related, but one of the big areas of research these past couple of years has been the infant microbiome. And today I'm going to talk about, what we did to really try to understand what's going on in the infant microbiome in the U.S. population and how that led to discoveries that eventually led us down this path to probiotic development. So over the past 5-6 decades, we've seen a significant rise in non-communicable diseases. These are chronic illnesses like allergies, asthma, and obesity. And these types of diseases are multifactorial. There's a genetic and an environmental component to it. However, over the past few years, we've also started to realize that there's this other very significant influencer of this disease progression, and it's the gut microbiome. Now, I like this figure that really shows how something that starts in the gut can start to accumulate systemically. So what we see here is on this side, we see this healthy gut microbiome. And what we have are these healthy microbiome influencers or microbiome influencers that are causing it to have a healthy microbiome. These can be medications, diet, and the environment. And in this case, everything's working well in the microbiome, creating these very beneficial metabolites. One of those example are these secondary bile acids. These, among other things, allow for niche protection, preventing opportunistic pathogens from coming in and setting shop. But they can also produce these other short-chain fatty acids found right below them, the butyrate, propionate, and acetates. And these are important because they do cause a localized change in the environment of the gut microbiome, but they also provide nutrition for those epithelial cells. And through passive diffusion, they can actually go into the lamina propria and start to interact with our immune cells. 90% of our immune cells are located in our gut. So there's a lot of interaction going on in here. What could happen in this example of dysbiosis is what we've done now is we've introduced antibiotics. These antibiotics have disrupted that microbiota. In this case, we're seeing this increase in proteobacteria. And this does a couple of things. One of them is that we are no longer producing these beneficial metabolites, the secondary bile acids or these short-chain fatty acids that are providing nutrition and immune stimulation. But we're also now producing these other metabolic byproducts that can be considered toxins. These toxins actually can go in and start to loosen up the tight junctions between the epithelial gut lining, causing essentially what's called a leaky gut, allowing other things to go from the lumen to the lamina propria. And as these gut metabolites and other things start to accumulate here, that's how it can enter the bloodstream. And once it enters the bloodstream, this is really how we start to get more of these systemic diseases start to emerge. What's interesting about the infant gut microbiome is that when mothers are pregnant in the placenta, there is no microbiome present there. You're essentially a clean slate. And there's really these two factors that significantly influence what your microbiome is going to look like early on. One of them is your mode of delivery, whether you're born vaginally or via C-section is going to have significant impact on what those initial pioneering microbes are. And then the other big contributor is your feeding type, whether you're being fed breast milk versus formula. That's going to have an impact on the localized environment and what those microbes now are feeding on. Now, what's interesting is that these changes that we can clearly see early on in the microbiome due to these factors start to disappear as an individual goes through toddlerhood and childhood to the point to where it starts to look more like an adult-like microbiome. And we can't really now tell who was breastfed or who was born via C-section. But what we do know is that at this very early stage between birth and infancy, there is a lot of interaction between the microbes in your gut and your immune system, fine-tuning that immune system. So this early imprinting that happens has these lifelong health outcomes in the individual. That's why even though at the end, everything in the microbiome starts to look the same, these changes have already happened. I'm just focusing in on breast milk right now. When we look at human breast milk, there's a lot of macromolecules in here. We see a lot of protein, a lot of fat, a lot of lactose, a lot of these things that are really going to be nutritious for the baby, allow it to bulk up, But we also see that the mother's producing a lot of energy in these other macromolecules called oligosaccharides, or these human milk oligosaccharides. And what's interesting about these human milk oligosaccharides is that unlike these other fats and proteins, they're not directly nutritious for the baby. Among the things that they can do is they can act as prebiotics for the microbes that are already in your gut. And this is just an example right here showing you of these different HMOs. So we got to remember HMOs come in many different flavors, and the types of flavors that are being produced are going to also be dependent on the mom's genome and genetic type. However, there are certain groups of microbes that do carry the genomic repertoire to be able to interact with these different flavors and break them down. In this case, one of the most popular ones are Bifidobacterium species. They can interact with a wide array of these human milk oligosaccharides, and in the process making these metabolites, these short-chain fatty acids that we talked about being able to modulate the immune system. However, even within Bifidobacterium, there are these more prolific individuals at being able to go in and break down a whole array of these HMOs. These are called Bifidobacterium infantis. They just have in their genome enough of these genes that allow them to interact with these HMOs, producing these secondary metabolites and change the localized environment. But what we've seen is that in non-Western populations, there's these high levels of Bifidobacterium that as we start to get into these Western populations, we see that these Bifidobacterium species, infantis species, are disappearing. So, these keystone species able to interact and break down a wide array of these mom's HMOs are starting to disappear. And that's kind of where our story began. What I'll talk about today is going to be a lot from this paper that we just published a few months ago. But what we did is we began the study called the My Baby Biome study. It was developed to deliver novel biomarkers for infant probiotic development, but also give us a better understanding of what's going on in the infant microbiome. We collected samples from individuals 1 to 3 months. None of these were fed any solid food yet. They were either breastfed, formula fed, or a mixture. And we did also collect fecal samples. Part of it is, we could talk about it later, maybe during lunch, but we did use those fecal samples to later on go in and isolate specific strains of interest. But once we isolated we collected these samples, we ended up with 412 samples with metadata, and we collected this metadata for 7 years. We have annual lifestyle questionnaires to evaluate atopy, obesity, and other health developments. Out of those 412 samples, 157 of these underwent metabolomics. And at the end, some of these follow-up experiments are from the longitudinal metadata, which 210 individuals were able to provide. This really became the largest US study to map the infant gut microbiome. And this is because we not only were able to collect samples from 48 out of 50 states, but we also try to make sure that our demographics that we collected were in line with the US population as far as getting the correct percentage of individuals born vaginally versus C-section, the feeding type that they're getting, ethnicity. We really tried our best to make it as representative of the US population as we could. And what we saw right away looking at it was that, as others had noted, 25% of infants completely lacked any sort of Bifidobacterium, and only 8% of our samples contain any sort of B. infantis present. What we also saw with that colonization of Bifidobacterium species in the infant gut is influenced by priority and niche effects. What I mean by this is if we start with this volcano plot right here, what we're looking at is the prevalence of certain microbes based on birth mode. And we see that individuals born via C-section, we see more of these Clostridium perfringens, those Enterococcus faecalis, these bugs that are kind of associated as being more opportunistic pathogens in individuals that are born vaginally. We see a lot of these Bacteroides. These in the blue are Bifidobacterium species. While they do have a higher likelihood of being present if you're born via vaginally, it's not a guarantee. Other thing that we looked at was how feeding influences the microbial community. So in this case right here, we're looking at breastfed versus not breastfed. And we could see that individuals that are breastfed, we have a higher abundance of kind of this bugs that we would expect as far as the Bifidobacterium infantis goes. However, we also see high abundances of these other bugs that are HMO consumers and also potential pathogens in breastfed individuals, Clostridium perfringens and these Klebsiella. This figure down here now is just breaking down the abundance of infant bifidobacterium based on the different feeding types, breastfed, mixed, or formula fed, and then the birth mode. And the main thing to highlight is while being breastfed and born vaginally does give you a higher likelihood of getting these important bifidobacterium species, this isn't a guarantee. And this is, this was really surprising to me because when we had our first child was around the time that we were starting to do these studies. You assume you breastfeed, you're born vaginally, you've done everything right, there's nothing else you can do, and you move forward, you're going to get all the correct bugs. But what we're seeing here is that that's not the case, right? This isn't the guarantee, and simply just breastfeeding isn't also going to always guarantee that you're going to get the right bugs, because you can get other bugs that are starting to occupy that area that aren't necessarily good for you either. And then this last one also just shows infant bifidobacterium and their effect on other bugs. And you can see that the presence of infant bifidobacterium decreases the likelihood of these C. perfringens bugs being able to colonize. Once we looked at the individual microbes, one of the things that we can then ask is, are there any natural clusters or types that start to emerge from our data? This is a PCOA plot. Each of these individual figures represents one individual's microbiome. There are tens of hundreds of microbes condensed into this two-dimensional space. The closer two of these are to one another, the more similar the communities are. The further apart, the more dissimilar they are. And what we could start to ask is, we take a probabilistic approach that looks at everybody's microbiome data and starts to look for patterns and say, can we start to bin individuals in particular patterns? Maybe it turns out that all of these individuals right here all tend to have high abundances of microbe A compared to everybody else. Maybe they belong in their own bucket. This other group of individuals have high amounts of microbe B, different from everybody else. Those deserve to be in their own bucket. And then these other group of individuals seem to be somewhere in between. And you can see how this is really just an agnostic approach to being able to ask, are there any microbiome patterns that emerge? And what we saw was that, yes, there was these three distinct enterotypes that emerge, the C1, the C2, and the C3. And one of the things that we're seeing is that, at least in the PCOA plane, what's driving these clusters are the abundances of infant bifidobacteria. We see very high abundances of infant bifidobacterium here, and it starts to disappear as we move away into that C3 cluster. And once again, this is just another way of showing you those three different clusters. The C1, a lot of, this right here denotes a pie plot of the abundances or the contributions of a phylum to that given community. And then the bar plot representing, the highest contributor as far as species goes in a certain phylum. What we see in the C1 is that it's largely driven by this Bifidobacterium breve abundances. We see that it's present in very high abundances. What we see in C2 is we start to get a higher amount of Bacteroida starting to emerge. But as far as Bifidobacterium goes, B. longum seems to be the largest contributor. And then finally, the C3 cluster, we see that it's completely dominated by Firmicutes and Proteobacteria. At the end, what we saw was that our C1 individuals constituted 24% of our samples. This is what we call healthy. It's just classic Bifidobacterium rich. And all these individuals, or this group of individuals, were really dominant and breastfed. Or was dominated by breastfed individuals. The C2 is one form of unhealthy. This one's enriched in Bifidobacterium longum and Bacteroida. And while I'm not showing the data here, we do have a large amount of adult samples as well. And when we looked at the similarity distance between these clusters and the adult samples, we saw that C2 cluster resembled that of an adult-like microbiome. So that's why we called it more of an adult-like microbiome. And then finally, the C3, this one is also unhealthy, a different type of unhealthy, enriched in these Firmicutes, Proteobacteria, opportunistic pathogens, and a lot of C-section dominant individuals. Next, what we want to look at was HMO utilization, antibiotic gene resistance, and virulence factors across enterotypes. So what you're looking at is a heat map. Red means that there's higher abundances of these genes relative to the other samples. And blue means lower abundances. First, we've grouped them by the cluster that a sample belongs to. So all the individuals here belong to cluster 1, cluster 2, and then cluster 3. And then finally, the last question that we asked were, who are the primary HMO utilizers here? Who are the taxa that are contributing the most HMO genes in that particular sample? In C1, we see that there's broad usage of all these different HMOs, and that B. breve seems to be the top HMO user. For C2, what we see is that B. longum is the top user of HMOs here, but you can start to see that there's reduced HMO cluster abundances within here, specifically between that H4 and H5 region. And then finally, the C3, what we saw was that Clostridium species, or Clostridium taxa were the top HMO consumers. We really start to see limited functional breath here, and we did see at least a subcluster that has this very high urease usage abundance. Other dysbiotic indicators that we looked at were antimicrobial resistance and virulence factors. And once again, what we see is that virulence factors are highest. Virulence factors and antimicrobial resistance genes are highest in the C3, followed by C2, and finally, they're the lowest at C1. We see that there's a loss of infant bifidobacterium associated with increased antibiotic resistance and virulence factors. So in this plot, you're just seeing the association between antimicrobial resistance genes up here, virulence factors, and the abundance of infant bifidobacterium abundances. And you see that there's a negative correlation here, suggesting that there is a protective association of the infant bifidobacterium, highlighting the potential importance of being able to shape a healthier microbiome. We also, when we first looked at the metabolomics of the infant gut microbiome, we did a pilot study and we found that what you're feeding the infant has a big impact on the metabolism. It was a very strong confounding factor. So then what we did for all our follow-up analysis is we said, let's just focus on breastfed individuals. So all of the individuals would be breastfed, but they're all coming from these different clusters. That way we can get a better understanding of what's going on in these communities. When we do that, we're essentially looking for associations, combination of ways, correlations, conditional independence, just ways of being able to make sure that these associations are strong. We end up with a whole list of what these associations are, and one way that we can start to really visually explore and try to understand these communities are through these network analyses. When we start to build these network analyses, each of these circles represents an individual feature. The lines represent their association. So black line means there's a positive association. These pink lines represent that they're negative associations. And when we do it with all our microbiome and all our metabolites that pass their statistical thresholds, we get this huge furball cloud. But within that furball cloud, we can start to ask, are there any strong clusters or communities that actually are very tightly clustered with one another, meaning that they're probably associated with a particular part of the pathway, a particular part of the ecology, that are tightly associated with one another, that are separate from everybody else. When we ask this question, one of these modules or subclusters that emerge is this one right here that I'm showing you. What we could see is that there's this very strong association between Bifidobacterium breve, our B longum, and our B infantis. And in one case, they're playing this very complementary role in producing immune regulation via metabolism. We see that all of these together are producing these important metabolites like thiamine, vitamin B, this 4-hydroxyphenolactate, and these indole-3 lactates. Especially the indole-3 lactate, very popular, very important one, because this particular metabolite has been shown to be involved in interacting with the immune system, both in vivo and in vitro, and having an impact on having a pro-inflammatory, anti-inflammatory response from the immune system. What we're also seeing is this other group of interactions right here. In this case, it's a negative association between B breve and B longum. In this case, it's suppressing potential pathogens like Clostridium perfringens and Enterococcus faecalis and these other bugs. And this along with the other results really started pointing us towards what constitutes a healthy microbiome, what are the bugs involved in a healthy microbiome. And from a lot of this is where we started to follow up with being like, okay, are there specific strains that are better at doing these than others? Are there specific strains that are better at interacting with each other than other strains? And this was a lot of the follow-up stuff that happened beyond the paper. Other things, as I mentioned before, is we did have follow-up questionnaires for these infants or for these children. When we got it from 1 to 3 months, at the time of the publication of the study, we had 2-year health outcomes from these infants. What we saw was in the 210 individuals at age 2, already over half of them, 54% of them, had already reported antibiotic use at some point, and 30% of them have already been diagnosed by a physician as having adverse outcomes. And adverse outcomes means they had asthma, allergies, As asthma analogies, these were part of the adverse outcomes. What we wanted to know is I mentioned these three different clusters earlier on and kind of what they're dominated by. We want to know, does being in a particular cluster have an impact in your risk of developing these adverse outcomes later in life? And that's what we see in this figure right here, is we saw that relative to that C1 cluster, that one that was very bifidobacterium rich, low in antibiotic resistance, low in virulence factor genes. Relative to C1, we see that C2 and C3 both had a threefold increased risk of an individual developing adverse outcomes by age 2. And this risk was similar in magnitude to early antibiotic exposure. And this was also very interesting because there's already a lot of data out there, suggesting that early antibiotic use does have an impact on these diseases or these chronic illnesses later in life. And what we're seeing as well is kind of where you are within those clusters also has an impact on those adverse outcomes. And then finally, what we also wanted to know is, are there particular features that are associated with adverse outcomes? This one was really interesting because we weren't focused on a particular pathway, like a KEGG pathway or a KZ pathway. We really did this other approach to try to explore all the dark matter, in which we grouped proteins together based on similarity, and then just looked at them based on these gene clusters, is essentially what we call them. So you can imagine we have hundreds of thousands of gene clusters, and we're trying to look for associations between those hundreds of thousands and this disease outcome. And after adjusting for multiple comparisons, we still get these individuals that are very strongly associated with adverse outcomes. And a lot of these proteins, when we did distance similarity checks to try to understand what they were, we saw that many of them are virulence factors. And some of these virulence factors are virulence factors that we know through literature to be involved in interacting with the immune system, such as directly interacting with the immune system in the host, such as LBS biosynthesis, and other of these were actually phage and transposon associated. And this was also very interesting because there have been a couple of studies that have come out very recently showing that certain phages and certain viruses being present in the early gut also has an impact on developing allergies and asthma later on in life. So in conclusion, this is the largest U.S. infant cohort to date. We were able to identify 3 distinct enterotypes, one that's bifidobacterium dominated, one that's high in Bacteroida, and one, this is more of the adult-like microbiome, and another one that's Proteobacterium and Firmicutes dominated. We do see a loss of these cornerstone microbes. 25% of infants lack any sort of detectable bifidobacterium species, and that keystone bifidobacterium, that B. infantis, was absent in 92% of these children. And the problem is also that in their absence, they start to be replaced by these other mucin utilizers, these HMO utilizers that also have the opportunity to use up mother's breast milk, such as that C. perfringens. We do see a functional consequence that results from this loss of bifidobacterium, disruption of the HMO utilization, increased antimicrobial and virulence factor genes, and a shift in the metabolome. There's clinical implications. By age 2, 30% of infants had allergies, asthma, eczema. Cluster C2 or C3, which represents 76% of this cohort, carried a higher risk of developing these. And we did see that B. brevi abundance is associated with a protective effect in our data. And we're continuing to monitor these children for the next 7 years to really understand further the long-term impacts, collecting their microbiomes and seeing what trajectory these microbiomes also take over time. And we do believe that early interventions targeting microbiome restoration can offer preventative potential against these non-communicable diseases. And with these results, that's how we came up with our new product that just came out in September. This is our symbiotic, and we do have ongoing clinical trials that are actually going to be completed by the end of the year. We just got finished up with our results looking at infants and toddler gut microbiomes, looking at the efficacy and tolerability of our symbiotic strain. For any of you that are interested, please visit us at persephone.bio. And with that, thank you. I don't know how much time, but I'm happy to answer questions. If not, catch me at lunch.